Welcome to the show. Man. Hey everybody, I'm Silas, and welcome to Psilocybology. Let's expand our mind. This is a series where we talk about the research on psilocybin, the psychedelic prodrug found in magic mushrooms. We'll be breaking down and discussing the scientific literature, one study at a time, from past to present. If you'd like to support the show, please make sure to rate, review, and subscribe to the podcast. Today's episode is on the article titled, The Influence of Hallucinogenic Drugs Upon In Vivo Brain Levels of Adenine Nucleotides, Phospho Creatine, and Inorganic Phosphate in the Rat, published in 1965 by researchers at the University of Glasgow. Let's start off today's episode with a brief bit of biochemistry so that we can understand what exactly is being studied by these researchers. An adenine nucleotide is simply a chemical that contains adenine, which some of you may remember is one of the 4 bases in our DNA, attached to a sugar molecule, in this case ribose, as well as one or more phosphate groups. When there's just one phosphate attached, we call that adenine nucleotide adenine monophosphate. When there are 2, we call it adenine diphosphate, and when there are 3, we call it adenine triphosphate. One thing that might be familiar sounding to those of you who may have taken biology or chemistry during primary or secondary school is the statement that ATP, aka adenine triphosphate, is the energy currency of the cell. Let's break that down a bit. ATP is referred to as the energy currency of the cell because it provides energy to drive forward a number of biochemical processes. The energy ATP contributes comes from the process that gets initiated by breaking off one of the phosphate groups attached to it. Strictly speaking, the energy donated doesn't come from the breaking of the bond itself, which requires energy to do, but rather the overall process that results from that bond being broken. For example, when ATP reacts with a molecule of water to form adenine diphosphate, aka ADP, and a single inorganic phosphate, this overall reaction releases energy that can then be used to drive another reaction forward. An analogy might be to think of a glow stick. Snapping the glow stick requires some amount of energy, but by putting in that energy, it initiates a process that results in the stick starting to glow. It's not the break itself that produces the light. However, that break is necessary for it to start glowing. Let's actually take a little bit of a closer look at the reaction we just mentioned, because in doing so, it'll put a lot of what these researchers examined into context. As we mentioned, one of the ways ATP provides energy is by reacting with water and breaking off one of its phosphate groups to produce ADP and an inorganic phosphate. This process can actually be repeated, whereby ADP reacts with water and breaks off another phosphate to form AMP and another inorganic phosphate. Now, because ATP is so vital for providing energy for our cells, there are systems designed to replenish our supply of ATP. One such process involves reattaching inorganic phosphates to AMP and ADP. Sometimes, however, this process alone doesn't replenish our need for ATP quickly enough, which is where phosphocreatine, the other chemical mentioned in the study title, comes into play. Phosphocreatine, also known as creatine phosphate, is, as it sounds, simply a phosphate group attached to creatine. What's important about phosphocreatine is that it can very rapidly transfer its phosphate group to ADP, thereby converting it to ATP. What's special about this process is that it can occur directly in muscle tissue, whereas other processes involved in creating ATP occur in mitochondrial cells. Thus, phosphocreatine is vital for replenishing energy during intense physical activity. For any more visual learners out there, I'll include a helpful diagram that illustrates the process we just discussed in the episode transcript, which you can find on the website psilocybology.org. So now that we've covered the importance of adenine nucleotides, phosphocreatine, and inorganic phosphate, let's jump into the study itself. Part of the rationale for this study is that the researchers noted in their introduction that, quote, amphetamine-like drugs and other types of antidepressives which cause behavioral stimulation have been shown to raise brain levels of ATP and lower brain levels of ADP. The reason for equating stimulant drugs with antidepressant drugs is because at the time, many stimulant drugs were being researched not as treatments for attention as they are often used now, but as treatments for depression, in part because they did, at least in the short term, improve mood. But anyway, given that prior research, these authors wanted to examine how hallucinogens appeared to influence levels of ATP and related compounds, in part because even back in the 60s, we were already seeing some evidence that hallucinogens may also have antidepressant properties. To do this, the researchers injected male albino rats with either drug solutions or saline solutions, and then killed, dissected, and had their brains extracted either 30 minutes, 1.5 hours, or 3 hours post-administration. In total, 10 different drugs were administered, each at 2 different doses. These drugs were cocaine, mescaline, psilocybin, bufotenine, LSD, JB239, hyoscine, atropine, 5-HTP, and 3,4-dihydroxyphenylalanine, also known as DOPA. Let's briefly go through those. Cocaine, as I'm sure many are familiar with, is a stimulant drug which greatly increases levels of serotonin, norepinephrine, and dopamine in the brain. As I'm sure you're also familiar with, mescaline, psilocybin, and LSD are 3 classic hallucinogens that we see compared quite frequently in this early line of research. If you're a regular listener of this podcast, you may also be familiar with bufotenine. Bufotenine has come up quite a bit in some of the earlier episodes, because it's very similar in structure to psilocybin and is the hallucinogen that can be found in the skin of some species of toads. Now, if you've really been listening since the beginning, or have bravely dived into some of the first few episodes, you might also recall JB329 is a drug we discussed in episode 6. JB329, marketed as Ditran, was developed during chemical weapons research. The goal of this research was to essentially develop non-lethal chemicals that could incapacitate their targets. JB329 works in part by blocking the action of acetylcholine in the nervous system, which is a neurotransmitter that, among many other functions, has a prominent role in muscle movement. During that episode, we noted that participants given JB329 experienced substantial mental confusion, which often manifested in such severe delirium that many participants were required to remain in the hospital overnight. Participants were disoriented, depressed, and described the experience as extremely unpleasant, if they were able to even remember the experience at all. Personally, I despise the fact that this drug was ever studied alongside drugs like psilocybin, because it bears no resemblance to classic hallucinogens. The reason for its inclusion, however, is because some of the prior research that found certain drugs increased brain levels of ATP was specifically with drugs that appeared to block the action of acetylcholine. That is also the rationale for including hyoscine and atropine, which are medications that both have anticholinergic properties, meaning that they also block the action of acetylcholine. Finally, 5-HTP and DOPA are precursors used in the biosynthesis of serotonin and dopamine, respectively. Although to be a bit more comprehensive, DOPA is also a precursor for norepinephrine and epinephrine. So as we've already discussed, the outcomes examined by these researchers were brain levels of AMP, ADP, ATP, phosphocreatine, and inorganic phosphate. Although those are the primary key measures, the authors also calculated 2 secondary outcomes. The first was the ratio of ATP to ADP, so just the concentration of ATP divided by the concentration of ADP. The rationale for including this measure is that the ATP to ADP ratio is actually incredibly important because it drives a lot of biochemical processes. Without diving too deep into the nitty-gritty details, there are certain processes in the body that can be regulated by changes in the ATP to ADP ratio, because this ratio acts as a sensitive indicator of how much energy is being consumed. As ATP is converted into ADP to provide energy for the cell, that ratio decreases. When ATP is replenished, the ratio increases. There are key proteins that monitor this change in energy status and can promote or inhibit biochemical reactions accordingly. The second calculated outcome is the total amount of AMP, ADP, and ATP. Since, as we've discussed a little bit already, ATP can lose phosphate groups to become ADP and AMP, and AMP and ADP can gain phosphate groups to become ATP, these nucleotides are largely recycled. If in the results we see an increase in ATP and a decrease in ADP and AMP, but no changes in the total amount of all three, this would support the conclusion that all of the increase seen in ATP comes from the conversion of ADP and AMP. I don't know the health implications for either an increase or decrease in this total sum, but perhaps it may indicate when there's some disruption to the natural process of recycling between AMP, ADP, and ATP, which I imagine could have consequences, but again, that's just my speculation. If anyone listening knows more about this, definitely let me know. Now that we have a full grasp of the methods, let's get into the results. Since there are so many drugs tested, I want to start by walking through what they found for psilocybin first, and then discuss how these results compared to the other drugs examined. In this study, rats were either administered 0.05 milligrams of psilocybin per kilogram of body weight, or 0.1 milligrams of psilocybin per kilogram of body weight. All comparisons are relative to rats administered a control saline solution. Starting with the adenine nucleotides, 30 minutes after administration of either dose of psilocybin, researchers found a decrease in ADP and an increase in ATP, with an accompanying increase in the ATP-ADP ratio. For the larger dose, they also found a decrease in AMP. No change was seen for the overall sum of AMP, ADP, and ATP. The researchers also observed an increase in phosphocreatine. No change was seen to levels of inorganic phosphate. When they looked next at the 1.5 hour mark, they found the same decrease in ADP and an increase in ATP. They didn't, however, see the same decrease in AMP or the same increase in phosphocreatine. Again, this was for both the 0.05 milligram per kilogram and 0.1 milligram per kilogram dose. Finally, looking at the 3 hour mark, the researchers only observed any significant changes for the higher dose. Specifically, they found again a decrease in ADP and an increase in ATP. This appears to be the most robust change after psilocybin administration out of the outcomes examined and given their dose ranges. Based on the increase in both ATP and initial increase in phosphocreatine, it does appear that this study indicates psilocybin increases the available energy in the brain. Before we compare this to other drugs, these findings have some interesting implications. First off, increases in brain activity appear to parallel increases in brain levels of ATP, which may be because as the amount of energy available to a cell increases, this can often act as a signal to increase energy utilizing processes. I also came across some provocative studies linking levels of either ATP or phosphocreatine to certain health outcomes. One such study found that substances which enhance levels of ATP can improve cognitive functioning. Another found that phosphocreatine appeared to be protective against ischemic injury, which is a brain injury caused by diminished or absent blood flow to a region of the brain. On the mental health side of things, there was also a study that found decreased levels of phosphocreatine in patients with severe depression. Now, if you also remember how earlier we discussed how phosphocreatine can be used to quickly replenish levels of ATP, well, there was a study that found that there is a, quote, failure to replenish ATP from phosphocreatine in patients with bipolar disorder. There was also a cool protocol published this year in 2019 for a planned pilot study to administer ATP and phosphocreatine in combination with the antidepressant fluoxetine, because the researchers believe it will significantly improve treatment outcomes. Now, if you follow me on Twitter, you're probably aware that I'm not a huge fan of the pharmaceutical industry, but I'll still be curious to see the results. Because if ATP and phosphocreatine do improve outcomes compared to fluoxetine alone, then that incidentally provides support for using an antidepressant that naturally raises levels of ATP and phosphocreatine, like psilocybin, for instance. Let's move on and compare the results from psilocybin to those of the other drugs administered in this study. There was one drug, albeit at a different time point after administration, that actually produced a completely identical pattern of results, and that drug was mescaline. Specifically, the results seen 1.5 hours after administering either 15 milligrams of mescaline per kilogram of body weight and 50 milligrams of mescaline per kilogram of body weight produce the same set of outcomes as seen a half hour after administering either doses of psilocybin. Since we know psilocybin is faster acting than mescaline, this makes sense and is honestly what we might expect. Now, I'll qualify that the authors didn't compare the magnitude of change between these conditions, or for any of the drugs, for that matter. So it's unclear if one of these two drugs produced a greater or lesser change than the other. However, both produced increases in ATP and phosphocreatine, as well as decreases in AMP and ADP, without changing the total sum of all three nucleotides. The next drug that seemed to have a very similar pattern of results was bufotenine. 30 minutes after administering either 5 milligrams per kilogram or 10 milligrams per kilogram of bufotenine, the only difference in response to that from psilocybin was that there was no statistically significant decrease seen in AMP. This may be only a relatively minor difference, however, since this decrease was only seen once, 30 minutes after administering 0.1 milligrams of psilocybin per kilogram of body weight. Now, the pattern of response seen after bufotenine actually matched that of LSD with one notable exception. Although at the half-hour mark the response to LSD and bufotenine were identical, the researchers found that 1.5 hours post-administration of both the 0.05 milligrams of LSD per kilogram of body weight and 0.1 milligram of LSD per kilogram of body weight, there was a significant increase in the total sum of AMP, ADP, and ATP. This increase was only seen for that 1.5 hour mark and was no longer significant at 3 hours post-administration. As I mentioned earlier, I'm not sure what the implications this has. On the topic of LSD, I also wanted to note that I found it odd the doses used for LSD were the same as those used for psilocybin, given that we know LSD is substantially more potent, by orders of magnitude. Normally, LSD is measured in micrograms, not milligrams. I'd be interested to see what the results would be if the researchers administered a more therapeutic range of LSD. Now, similar to LSD, hyoscine produced largely similar patterns that we've already discussed, in addition to the increase in the total sum of AMP, ADP, and ATP for that 1.5 hour mark. Hyoscine, however, stood out because it was the only drug to produce a significant rise in levels of inorganic phosphate. So far, we've seen 2 instances of an increase in that total sum outcome, once for LSD and once for hyoscine. One thing that I found interesting is that at both doses and at 2 of the 3 time points, JB329 increased the total sum of these 3 nucleotides. And then somewhat paradoxically, JB329 also produced a significant decrease in this sum at the 3 hour mark, which seemed to be driven by reductions in AMP and ADP without any parallel increase in ATP. This to me strikes me as a potentially key distinguishing feature of JB329, because it is the only drug that produces a decrease in this calculated outcome. The last few drugs all stood out a bit from the rest, partly due to a lot fewer significant responses in general, which I'll note may absolutely be a result of being given too low of doses or maybe very rapid durations of action. That sort of goes as a limitation for the entire study. Take each of these comparisons with a grain of salt, because they all vary in terms of their therapeutic dose ranges and how quickly they move through the body. Anyway, cocaine was interesting because it appeared to only produce a significant increase in ATP. It did appear that there might have been some reductions of ADP, but this decrease was not significant. There was also no increase seen for phosphocreatine. One limitation that's specific to cocaine is that they only examined rats 1.5 hours after administering cocaine, so they did not look at the 30-minute or 3-hour mark. Cocaine is pretty fast-acting, so missing that 30-minute test I think is actually very crucial. Atropine was actually pretty similar to what we've seen with increases in ATP and decreases in ADP and AMP. But there was no change to levels of phosphocreatine for any of the trials. Finally, there were very few changes seen after 5-HTP and DOPA. That said, both of these drugs were the only ones that appeared to increase levels of AMP. No changes were seen to the other two nucleotides. Overall, the authors noted that one of the most consistent findings was an increase in ATP, and they believe that such an increase may be responsible for producing some of the antidepressive and stimulating effects of these drugs. One last note that I want to make is that the study focused on acute changes in these outcomes after drug administration. I'd be interested, however, in how chronic use of these substances may influence chronic levels of these outcomes. Are the increases sustained over time after repeated administration? Do any of these drugs produce initial increases followed by a depletion of these compounds, which was potentially hinted at in the case with JB329? Part of this interest comes from a study I came across that found reductions in phosphocreatine in individuals who use methamphetamine compared to non-users, and that the decrease was correlated with the amount of methamphetamine use. This would be an important outcome to study so we can better understand the potential benefit or harm of these drugs. And with that, that's it for today's episode. As always, I really hope you enjoyed the show. If you have any questions, feedback, or ideas for the podcast, please let me know. You can find out all the ways to reach me on the website, psilocybology.org, where you can also find the full transcript for each episode if interested. And links to the studies I referenced are embedded in the transcript. Thank you all for listening, and I'll talk to you next time.